Hello, we are Group 2. I am Alyssa.
I'm Cynthia.
I'm Daniela.
I'm Dean.
I'm Jasmine.
I'm Maritza.
Our general purpose was to determine whether the topsoil or the bottom soil had more microbes. Our question was, does the farm soil have more microbes depending on how deep it goes? UV rays, where our hypothesis was the UV rays, does it kill more microbes closer to the surface? And then our prediction was the bottom soil contains more microbes than the topsoil.
Okay, our control variables were the location, the moisture, the association with the plants and animals, and the temperature. Our experimental variables were the light exposure or UV rays.
So these are our results. Here you can see the topsoil and the SDA the SDA agar we use to grow the fungus. And then over here, the bottom soil we use AIA, and this is where you can see we grew the most microbes. Here you can see the fungus that we grew on the topsoil. This is for the bottom soil. And then these are the bacteria that we grew for topsoil and the bottom soil. And then we also had some gram negatives for the top bacteria, and then the gram positive for bacteria too.
Our results on our final microbe concentration, we had for our topsoil, we had 1.25 multiplied by 10 to the 6th power for our bacteria. And for our fungi in topsoil, we had 2.7 multiplied by 10 to the 6th. And for our bottom soil, we had 1.77 multiplied by 10 to the 6th for our bacteria. And for our fungi on our bottom soil, we had 1 multiplied by 10 to the 4th.
We have the differences between the top bacteria, top fungi, bottom bacteria, and bottom fungi. For the top bacteria, we have gram positive and negative. And the morphology is different. It's cocci and bacilli. And we see the colonies on the plate. The top bacteria one is orange circular, and the top bacteria 2 is white circular. So it's different, definitely different species. Another one is the top fungi. We grow the white circular colony, which is different than the bottom fungi. The bottom fungi is the black rhizoid colonies. And for the bacteria on the bottom side, we have gram negative and positive, and we saw the coccobacilli on the bacteria 2. That's definitely the different species as well.
Our conclusions were that the results rejected the hypothesis, the UV rays have no impact on the results. And the conclusion based on the prediction, the topsoil contained more microbes than the bottom soil, and there were different species of fungi and bacteria between the top and the bottom soils. What could be done in the future? In the future, we could conduct an experiment that has a controlled variable of direct and indirect sunlight.